I would like to extend you a very warm welcome to my class today on the topic of sterility testing with the help of direct inoculation technique. This was the topic which was under Pharmaceutical Microbiology with a code of BP303T for the B.Pharm third semester students for our university, APJ Abdul Kalam Technical University. Now, as today the entire world is moving towards outcome based education, so let me just give you an outcome of the class today. At the end of the class today, you will be in a position to know how you will be in a position to determine whether the substance which you have sterilized has been properly carried out, and you can recommend it for further use. That is, the students will be able to have a clear idea about whether the sterilization process which they have adopted has been successful in removing the microorganisms. I am Dr. Abhijit Majumdar, Professor and Director of Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. Now, the term direct inoculation. Now, as I have told you, let me quickly recapitulate that sterility testing is basically done by two methods. One was by the process of membrane filtration technique, and the second one was by the process of direct inoculation technique. Now, membrane filtration technique, as we know that we have to convert the liquid, a solid into a flowable form, and then you have to carry out the process of membrane filtration. In case of liquid, we can do it as such, just filter the liquid through the membrane filter, and then the membrane filter is cut down into different segments, or if, as I have shown you in the last video, that if we are carrying out in a set of 3, one we can put it in the thioglycolate media, the other one which we can put in the alternate thioglycolate media, and the third we can put it in soybean casein digest broth. So, whenever we will be putting it in the aerobic, anaerobic and a fungal media as stated, it will create a turbidity. Moment turbidity has been created, it means the substance which you are testing has not been properly sterilized. The other technique which we also use to determine whether the substance has been properly sterilized or not is what we are calling as direct inoculation. Now, here the samples need to be transferred directly into the media tubes to assess the presence of any viable organism. And if the media in the media tubes are converted turbid, or it loses its transparent character, it means that the substance which you are testing has not been properly sterilized, and hence it will not pass the sterility testing. Now, this is commonly used for solids, where you have to dilute it tenfolds, and In case of liquid, if the volume is small, you need to dilute it 100 times with the help of any diluents. You have to use or take the help of an emulsifying agent if the preparation is water insoluble. Particularly, we are talking of the oily preparation where we need to add an emulsifying agent and then put up directly into the medium. Now, let us see what or where these direct inoculation techniques are commonly used. Direct inoculation techniques, they are used for powders which are water soluble in nature. We are using for liquids which are water miscible in nature. We are using for various solid substances like gauze, cotton, catguard, syringes, and so on. We have to maintain one common precautions which I have told you in the earlier classes also, that is, whenever you are carrying out these tests for sterility, you have to be very sure of that the area where you are carrying out this test is absolutely free from microorganisms. That is, the media which you are using, the glass apparatus which you are using, that mandatorily needs to be bacteria or fungi free, that is, free from microbes. The environment where you are carrying out this test, it mandatorily needs to be aseptic. A question may be coming up in your mind, why are we repeating this again and again? I am repeating this because if the environment where you are carrying out this test is not properly sterile, you will not be in a position to know just by seeing the turbidity that the turbidity has been caused by the nature or the environment where you are carrying out this test, or it is due to the substance not properly sterilized, that is your sample. That is, you will not be in a position to justify that the turbidity has been created by the microorganism present in the environment or the microorganism present in the sample. Just to rule out that the turbidity has been created by any factors related to the environment, we have to carry out this test in a very aseptic environment. That is, high precautions need to be taken in order to avoid all sorts of accidental contamination. Now, as I have told you that it is a process which has been recommended by Indian Pharmacopoeia, and all the industries, all the pharmaceutical industries across the country, they have to follow it stringently as a guideline, because depending on your test, depending on your report, the product will go into the market. If the product is having less than 1 ml, you need to add the entire contents of each container. But if your product is having a volume of 1 to 40 ml, you have to add half the content of each container, but not less than 1 ml, and preserve the residue. If the product volume for that liquid preparation is greater than 40 ml, but less than 100 ml, then the minimum quantity to be used is 20 ml. However, if the product is having a volume, final preparation is having a volume of more than 10 ml, more than 100 ml, in that case you have to take 10% of the volume of the final container, but it should not be less than 20 ml. So, these guidelines, they are laid down as per Indian Pharmacopoeia. Now, if the preparation which you are testing is basically an antibiotic in a liquefied form, the minimum quantity to be used is 1 ml of the solution. Now, there are many preparations which are non-antibiotic in nature, which are not antibiotic. If the preparation is such, now here if the preparation is antibiotic in nature, we need to deactivate the compound, we need to mask that compound so that it should support if there is a presence of microorganisms. If it is of any compound which you are testing is basically having an antimicrobial action, you need to block, you need to mask the antimicrobial property by any method, it may be a chemical or an enzymatic method, so that the compound should not exhibit any antimicrobial action against the contaminant. Apart from antibiotic solutions, all other preparation which are soluble in water, or in case of isopropyl myristate, you need to take the whole content of each container, but the quantity should not be less than 200 milligram, minimum that quantity must be present. If you are going to test various insoluble preparation or cream or ointment, we need to suspend or convert it into a suitable emulsion with the help of emulsifying agent. That is, insoluble preparation, cream, ointment need to be suspended or emulsified with the help of emulsified agent, and IP recommends that use the content of each container to provide not less than 200 milligram as told earlier. Now, as far as the solid is concerned, if you are testing a solid where the package is less than 50 milligram, you have to take the whole content of each container. If the quantity of the container ranges from 50 milligram to 300 milligram, if the content ranges from 50 milligram to 300 milligram, you have to take half the content of the container, but please take in mind that it should not be less than 50 milligram. That is, if the tablet or if the solid which you want to test, the quantity per container is 50 milligram to 300 milligram, half the content of each container has to be taken, provided that you are taking a sample and that sample should not be less than 50 milligram. If the quantity per container is 300 milligram to 5 gram, Indian Pharmacopoeia recommends that you have to take 150 milligram during the test, and if the solids they are having greater than 5 milligram, at least you have to take 500 milligram of the tablets or 500 milligrams of the solid. Depending on the final amount which is present in the container, we have to select the minimum quantity to be used during the test. So, we have discussed about the solids, we have discussed about the liquids. Apart from solids and liquids, there are various medical devices, there are various substances which can be tested with the help of this direct inoculation technique. Let us see what are they and how to evaluate. We can evaluate the catgut or other surgical appliances which are commonly used for veterinary use. If the substance which you are testing is of the nature of a catgut or any surgical appliances for veterinary use, take the strand, cut it into 3 different sections, and each section should be of 30 centimeter dimension. That is, if it is a catgut, if it is a surgical appliances, we need to cut up into 3 different sections, and each section must be of 30 centimeter dimension. At the end of the class, I will be showing you a video where you will be seeing how aseptically, how carefully in a controlled environment we are taking a sample and we are cutting it into 3 sections before directly inoculating into the media. Apart from catguts, we are in a position to check surgical dressings. We are in a position to test cotton as well as gauges. We have to be very careful whenever we are doing the test for cotton and gauges, because very frequently we apply this cotton in an area where we are having an injury. Therefore, the cotton and gauges which we are using must not contain any microbes. We should be very sure that it is aseptic. And the minimum quantity which we have to take from that package is 100 milligram. That is 100 milligram we need to directly inoculate into the media. Media again, a bacteriological medium, that again are of 2 kinds, one is aerobic, another one is anaerobic, as well as we have to take soybean, casein, digest broth as a fungal media. Apart from catgut, surgical dressings, cotton, gauges, we have to take sutures or any individually packed single use materials. Once you use and discard it out. If this is the nature of the substance, sutures or any substances for single use, you have to take the entire device. You have to take the entire device, and all other medical device, for that you need to take the entire assembly and cut it up into small pieces, and those pieces are to be immersed or dripped into the media. Next, we are coming to parenterals. For parenterals, if the batch size, see, whenever you are taking a sample, the sample should be so taken that it should represent the correct behavior of the batch. It should not be that you are taking a batch, the batch size is so small that it is not giving a proper result. That is, by checking that batch, you was telling that my product is sterile, but whenever it is going into the market, it is saying that your product is actually unsterile. Sampling needs to be so done that it should produce a very reproducible, validated results. So, for parenterals, if the quantity is not, the batch size is not more than 100 containers, you need to take 10% or 4 containers, whichever is higher. If the batch size is more than 100 containers, but less than 500 containers, IP recommends you need to take 10 containers straight away. If the batch size is more than 500 container, in that case you have to take 2% or 20 containers, whichever is less. So, if the batch size is more than 500, it indicates that you have to take 2% or 20 containers, whichever is less. If you are taking these guidelines for the minimum quantity of for test, the result which you will be getting will be reproducible and correct. For large volume parenterals, the same criteria goes. For large volume parenterals, we have to take 2% of the batch size or 20 containers, whichever is less. If the powder is of antibiotic powder, if it is a pharmacy bulk package less than 5 grams, you need to take 20 containers. If it is a pharmacy bulk package of 5 grams, you need to take 6 containers. If more than this, that is, if there is a bulk and blend, then you have to take according to the bulk solid products. Now, we are coming to the next category of product, which is basically the ophthalmic preparation and non-injectable ones. If the product is of the nature of ophthalmic, that is to be applied in eye, or non-injectable category, and if the batch size is having less than 200 containers, you need to take 5% of the batch size or 2 container, whichever is higher. 5% of the batch size or 2 container, whichever is higher. Because more will be your sample size, greater is the chance of getting the results correctly. If the final batch size is 200 or more, if the final batch size is more than 200, you have to take 10 containers minimum. You have to take the control tubes, sterile nutrient media, ideally it should be sterile, no turbidity should be produced. If turbid means the media has not been properly sterilized. Sterile nutrient media plus organism, ideally it should be turbid because organism should be growing. If accidentally it is clear, it means the media is not nutritionally full or supplemented. If test tube number 3, sterile nutrient media plus test substance, if the test substance is sterile, the media should be, the entire test tube should be sterile. If it is turbid, it means the test substance has not been properly sterilized. Test tube number 4, sterile nutrient media plus test substance plus organism, it means ideally it should be turbid, that is the organism should grow in presence of the nutritional media. But if accidentally it is not growing, it means that the test substance has some antimicrobial property which is preventing the growth of the organism, and hence the media is clear. So, ideally test tube number 4 should be turbid. Now, let us move on to the interpretation of the results. Now, after we are putting it in the media, if there is no growth, it means the batch has passed the sterility test. If there is growth, you cannot reject. You have to do a retest and see whether there is microbial growth or not. If there is no microbial growth on retest, the batches passes the sterility test. However, if there is a growth, you need to see what type of organisms it is there. If the organism of this retest and the previous one is same, you reject the batch, telling that it fails to pass the sterility test. But if the nature of organism is different, that is the first one and the second one, if the organism nature is different, you have to do a second retest, taking the double number of samples, and in that case, if in that retest, if there is no growth, the preparation passes the sterility test as per IP, but unfortunately, if there is growth, the preparation fails to pass the sterility test. So, during or end of the incubation, if no growth, the test passes. If growth is observed, preserve it, repeat the test. If no growth occurs, test passes. If growth occurs, compare the first with the previous one. If non-distinguishable, that is if they are not different, it means there is microorganism, the test fails. If it is distinguishable, repeat the second retest with twice the number of samples. If no growth, the test passes. If growth, the test fails. You will need to follow, you need to refer to the Indian Pharmacopoeia and United States Pharmacopoeia for this particular topic. Now, I will be showing you two videos which will give you an idea about how it happens in an actual industry.
Sterility samples may be tested by membrane filtration or direct transfer, depending on the type of sample that is submitted for testing. The sample packaging is opened in an ISO Class 5 hood. Samples may need to be manipulated in order to expose all surfaces to the test media. This manipulation may include cutting the sample and removing caps and plungers. The samples are aseptically transferred to the appropriate volume of test media. The sample is fully immersed and then incubated for a minimum of 14 days.
Class today, I hope you have an idea about how this direct inoculation technique is followed in the pharmaceutical industry. Thank you very much for your patient hearing. Thank you.